Hello everyone, as we already discussed about prokaryotic cell in brief, in this video we are going to cover it in detail. As we all know, these lack true nucleus and membrane bound organelles. These are smaller cells and multiply more rapidly than the eukaryotic cells. Even though they vary greatly in shape and size, it has a similar cellular organization. These are some of the examples of prokaryotes like bacteria, blue-green algae, mycoplasma, pleuropneumonia-like organisms. As we all know, these are smallest organisms and lack cell wall, which are pathogenic to human beings and animals. Example to this is Mycoplasma pneumonia, which causes a typical pneumonia, which is an abnormal one and requires special treatment for its cure. Here they given 4 basic shapes of bacteria. Let's discuss it in detail. Bacillus, which is rod-shaped bacteria like Mycobacterium tuberculosis, which appears pink colored long slender bacteria and causes tuberculosis and affects majorly lungs. Coccus bacteria, which is spherical in shape like Staphylococcus, which are arranged in grape-like clusters and appears purple violet color while Gram-Stein procedure and causes infections like skin allergies and hospital acquired infections. Streptococcus, which are arranged in chains, and these are also gram positive bacteria, causes infections like tonsillitis, pharyngitis or sore throat as major infection. Vibrio, which is comma shaped bacteria and causes infection cholera, which has rice watery stool as major symptom. Spirillum, which is spiral shaped bacteria. Example to this is Spirochates bacteria, which causes syphilis and shows spiral rotational movements while seen under dark field microscopy in laboratory. Let's discuss cell structure of prokaryote. It contains cell wall which surrounds the plasma membrane, which is selectively permeable in nature, so it interacts with the outside world. It contains fluid matrix which fills entire volume of the cell is known as cytoplasm. It contains nucleus which is not well defined, and the genetic material of the prokaryotic cell is naked because it is not enveloped by any nuclear membrane. It also contains genomic DNA, which is single circular DNA. Outside to the genomic DNA, it contains small circular DNA, which is known as plasmid DNA, which confers unique phenotypic character to the bacteria like resistant against antibiotics, etc. The plasmid DNA, which is mainly used to monitor bacterial DNA transformation, it is the one of the mechanism which is used in horizontal gene transfer. Here DNA transformation is the process where the cell takes free DNA molecule from other bacterial cell and incorporates into its own genome. It is mainly used in genetic engineering and while doing gene therapy. The prokaryotic cell also has inclusion bodies, which are unique forms which are present only in prokaryotes, and it also contains mesosomes. These are specialized differentiated forms and infoldings of the cell membrane. These are characteristic feature of prokaryotes. Here none of the organelles which are found in prokaryotes are present in eukaryotes except the non-membranous organelles like ribosomes. Let's discuss cell envelope and its modifications. Bacterial cell composed of 3-layered complex cell envelope which consists of glycocalyx followed by cell wall and cell membrane. Here coming to the glycocalyx, which is an outermost layer which is made up of polysaccharide macromolecules differentiated into two types based on its composition and thickness, which forms loose sheath around the bacteria as which is known as slime layer, and if it forms thick or tough capsule, it protects the bacteria from host immune system as it is sticky or gummy in nature. Coming to the cell wall of the bacteria, here the cell wall is a rigid structure which is made up of peptidoglycan layer. This is the reason if we use antibiotics like penicillin group drugs, amoxicillin or ampicillin for some of the bacterial diseases, these antibiotics acts by preventing cross-linking of this peptidoglycan strands causes cell lysis of bacteria which leads to death of the bacteria and shows bactericidal effects. Here, the cell wall functions as determines the shape of the cell and provides strong structural support to the bacteria by preventing the bacteria from collapsing. The cell wall is different in both gram positive and gram negative bacteria. Let's discuss about plasma membrane of the bacteria. As we all know, it is selectively permeable membrane, so it interacts with the outside world. It is structurally similar to that of eukaryotes. Here, mesosomes, which are specialized membranous structures of the plasma membrane, these are formed by extensions of the plasma membrane into the cells and forms in the form of vesicles or tubules, which helps in DNA replication. These mesosomes, which helps in formation of cell wall, DNA replication, as it consists of enzymes, it also helps in secretion processes. It increases the cell surface area of the plasma membrane. Let's discuss about flagella. These are filamentous extensions from the cell wall. This is made up of a special protein known as flagellin. These flagella, which helps in movement of bacteria, it is composed of three parts: filament, hook and basal body. Filament is the longest portion as it extends from surface of the cell to the outside. Let's discuss the flagella with diagram. This is the structure of flagella. It is composed of three parts, that is filament, hook and basal body. These basal body, again it consists of outer rings and inner rings. Filament is the longest portion of flagella where it extends from surface to the outside. Let's discuss the arrangement of these flagella and different bacteria with examples. Here, the flagella is differentiated into four types based on its arrangement. If the flagella is present at both ends, it is known as amphitrichus. If the tuft of flagella is present at one or both ends, it is known as lobotrichus. If the flagella is arranged all around the cell, is known as peritrichus, which we will see in Salmonella typhi bacterium. Let's discuss about pili, which are elongated tubular structures made up of special protein known as pilin. Even though they do not play a role in movement of the bacteria, it helps in adhesion of the bacteria to the surface of host cells, and it also helps in DNA transfer while conjugation, which is also known as sex pili. Here, fimbriae, which is small brittle-like fibers which sprouts out of the cell. It also helps the bacteria to attach to the host organisms and rocks in the strains. Let's coming to the fimbriae. These are small brittle-like fibers which sprouts out of the cell, which also helps the bacteria to attach to the host tissues. In some bacteria, it contains membranous extensions which contains pigments in them, which extends into the cytoplasm of bacteria known as chromatophores. Let's discuss about ribosomes and inclusion bodies in the prokaryotic cells. The prokaryotic ribosomes, which are cellular organelles responsible for protein synthesis and translating mRNA into specific sequence of amino acids. These are non-membrane bound organelles which are associated with plasma membrane of the cell. Coming to the size of the ribosomes, which are 17 to 20 nanometers. Here, several ribosomes with single mRNA, it forms polyribosome, which is also known as polysomes, helps in translating mRNA into proteins. Coming to the structure of prokaryotic ribosomes, these are 70S ribosomes which are present together as two subunits. One is larger subunit which is 50S and smaller subunit which is 30S. These are part of 70S ribosome. Here S stands for Svedberg unit which measures sedimentation rate. Coming to the inclusion bodies, these are also non-membrane bound organelles which are reserve material in prokaryotes which are stored in cytoplasm of the bacterial cell. Example to this are synophysian granules, phosphate granules and glycogen granules, etc. In some, these are present in case of viral infection because these are the sites of replication which are present as viral inclusion bodies. Example to this is Negri bodies in case of rabies virus which is caused by rhabdovirus. It composed of cytoplasmic proteins. Viral particles are cellular components of the virus. Coming to the gas vacuoles, these are gas-filled structures which helps the prokaryotic bacteria to regulate their position in aquatic environments and provides buoyancy to the prokaryotic bacterial cell which are present in cytoplasm of the bacteria. That's all for today's video. We'll meet again in my next video where I'm going to explain about eukaryotic cell. Okay? If you like this video, please like, share and subscribe to my YouTube channel.